I hope you're ready to hear from Dr. Allison Bradbury, Principal Investigator and Associate Professor at Nationwide Children's Hospital, who will break down how researchers prioritize conditions to study and explore the role patient groups have in shaping therapeutic strategy. Please feel free to submit questions throughout using the Q&A feature. They will be answered after the presentation as time allows. Also, before starting, we want to poll the audience. To access the poll, please locate the Q&A polls tab, which is to the right of the video player, and select poll questions. We are asking you to answer, where is your organization currently in its cell or gene therapy development journey? Thank you.
I'm really excited to be here today for the second annual Empowering Patients Summit. And today I'm going to be speaking with you about strategic starting points for cell and gene therapy research. So Dr. Stephen Gray just did a really great job of explaining that our DNA is transcribed to RNA, which is then translated to our functional protein. He also explained that these are read in a very important 3-letter code. Just one misspelling in this 3-letter code, whether it be an extra letter, a missing letter, or just a misspelling of the letter, can really alter the way that the transcript is read, and this can alter how a protein is made. The protein can be completely degraded, the protein can be shortened, or the protein can be misshapen and altered its function. Once we know how a molecular change alters protein structure and function, the next big question is, can we fix this gene defect? And if so, what is the best tool? Dr. Gray also introduced this idea that different molecular changes alter your protein in different ways. So a change in your DNA may result in what's called a loss of function, in which your protein structure and function can be absent, shortened, or degraded. And in this case, your therapeutic strategy might look like gene addition, providing a healthy, functional copy of a gene. Or he also explained that all-important junction between an intron, the removed part of your DNA sequence, and the exon. These type of changes in your DNA, which might be referred to as a splice site defect, may be especially amenable to modulation by an antisense oligonucleotide. In contrast, there are certain molecular changes that result in the gain of function, where there might be an accumulation of a protein resulting in toxicity. And these need a completely different therapeutic strategy, where you might need to knock down a gene to reduce the amount of protein being made. Regardless if a molecular change results in loss or gain of function, you may be able to use gene editing, which are these little molecular scissors, to change a specific molecular defect. Dr. Gray did a great job introducing each of these therapeutic strategies. So today I'm not going to go into further detail about the specific strategies, but instead I'm going to focus on this roadmap from disease characterization to therapy development, so that we know what our strategic starting points are in our gene therapy programs. First, what's absolutely important is that we have a really complete understanding of the disease mechanism. We really need to know what organ systems are implicated in disease, which cell types within those organ systems, and even the subcellular organelles and pathways. If we don't know what we need to target to correct, then we cannot drive our therapy to the appropriate cellular targets. Additionally, we might have off-target effects of our therapy if we drive it to cells that do not need the therapy. We also need model systems, whether these are in vitro, which are these cells in a dish, or in vivo model systems, which are most commonly mouse models. We use these model systems to understand the disease mechanism, but then they're also critically important in evaluating different therapies for safety and efficacy. Once you have an understanding of your disease mechanism, and you have model systems available. Then, if your disease is likely amendable to a genetic therapy, then this can be the appropriate time for therapy development. You might be developing multiple different types of a therapy concurrently. For example, evaluating a gene replacement therapy in comparison to an antisense oligonucleotide therapy in comparison to a gene editing therapy. And or you might have multiple iterations of one therapy. For example, as Dr. Gray mentioned, trying different promoters in your gene replacement therapy to target different cell types and or expression levels. Once you have your model systems and your type of therapy modality, you then perform efficacy and dosing studies. This is attempting to show that your therapy has benefit in the disease context and also establishing a dose range in which you reach therapeutic benefit but do not hit toxicity. After you've established efficacy and a dosing range, then you must move into safety and tolerability studies. These are typically done in healthy animals to make sure there's not any unwanted impact of delivering your therapy. After all these types of studies are done, then you can potentially move to the clinical trial stage. And this does not even detail all of the manufacturing stages of drug development and interactions with regulatory agencies. This pathway is really going to differ for each treatment modality. For example, antisense oligonucleotides likely won't require an in vivo disease model. For example, if you're trying to modulate one of those splice site defects, those often are not conserved between mice and humans. Therefore, the mouse model may not be valuable. So your primary efficacy testing may be done in cell culture, while you would still need healthy animals for safety and tolerability studies. This pathway also differs for each disease. It's really important to know what your starting point is. How much work has previously been done to understand the mechanism of disease, and do model systems already exist? So these are some really critical questions that will determine your roadmap of disease characterization and therapy development. So I'm just going to hit on each of these stages briefly. So for example, there have been a lot of advancements in vitro model systems. Historically, we used pretty simplistic 2D or carpet culture in cell culture model systems. However, recently you might have heard of these organoid models, which are more sophisticated, three-dimensional kind of organs in a dish that better recapitulate some of the disease phenotypes. These model systems utilize cells that are isolated from blood and or skin cells. These are then reprogrammed and characterized into induced pluripotent stem cells, and then those stem cells can then be developed into these more advanced organoid systems. So for example, if you do not fully understand the mechanisms of disease using in vitro model systems, and maybe even these more sophisticated organoid model systems would be a valuable starting place for your development of a gene therapy strategy. But this is a pretty specialized process to make these stem cells and organoid systems. So you would need a specialized strategic partner for this part of your development. Other programs might need generation of an in vivo model system. As I mentioned, most commonly, these are mouse models. Historically, we used what are called gene knockout or null mouse models. However, these really don't recapitulate human disease, and oftentimes they are not compatible with life because the protein is so crucial in development. So there are more specialized mouse models, such as conditional knockout mouse models, which will express a protein in only certain cell types or times in development. And or now with the advancements of CRISPR gene editing, we can make mutation-specific knock-ins of patient-specific variants, and these can allow us to make mouse models that really mimic a specific patient variant. So again, if your step in your development of a gene therapy would require an in vivo model system to be generated, then you will need to determine a strategic partner. And this very well may be different from a strategic partner that would be necessary for generation of in vitro model systems, such as organoids. Of course, the reason that we're trying to develop these model systems, whether in vitro and or in vivo, is really to move forward with testing of different genetic therapies. And again, as we discussed, there are so many different therapeutic strategies depending on the type of molecular change and how it alters your protein structure and function. So if you're moving forward with a gene replacement strategy versus an antisense oligonucleotide or a gene editing strategy, your strategic partner might be different because they need different expertise in designing, manufacturing, and testing these therapeutic strategies. For the safety and tolerability studies, once you've developed and tested a gene therapy, these will really vary widely depending on your therapeutic modality. For example, if you're giving an AAV-mediated gene addition therapy compared to an ASO, compared to a gene editor, the safety studies will look different. They will also vary widely depending on if there's any precedent of that therapy. For example, if you're using a tried and true AAV9 capsid that's already been in an FDA-approved drug, then they may not require testing in non-human primates. However, if there is a novel capsid that's never been in humans, then the FDA may require large animal studies. The FDA will provide guidance on proposed safety studies. Some of these already exist as written guidance. For example, the FDA has provided written guidance on rat toxicology study for antisense oligonucleotides. However, for most really novel therapies, the FDA will need to look at these on an individual basis. Moving to the clinical trial stage of your therapy development, again, that really is another decision point on a strategic partner. Many hospitals can have what's called a center for excellence. So, for example, if your disease is in the category of a leukodystrophy, then your clinical trial site, you might want to be looking at places that have a leukodystrophy center of excellence. And then there are also places that have focused gene therapy centers. And it's really critical when you're delivering specific types of therapy that there's expertise in those. Because gene therapy really comes with unique challenges in both delivery and monitoring. So it's really critical that the gene therapy is being delivered in a place that is experienced and equipped to handle these unique delivery and monitoring challenges. I often get asked a question about, clinical trials, about this N of one or N of few, or a more traditional phase 1-2 clinical trial design. And again, this really is going to differ compared to your therapy type. So for those true individualized or personalized medicines, such as an ASO for a specific splice site variant or a gene editor for a specific variant, those are true N of one type trial design. In contrast, if you're giving a gene replacement therapy, where you're replacing the entire gene and therefore patients with mutations throughout the gene may be amenable to that therapy, then that type of trial could be an N of one or an N of few or a more traditional phase 1-2 clinical trial design. This also is determined by the disease prevalence or incidence. Do you have enough patients to enroll a larger trial, or are there just one or a few patients that have ever been identified with this disease? And the other factor is really the viability for eventual market approval, because the way to, of course, get access for a drug to the largest population is for it to be an FDA-approved drug, and that can only happen after following through the traditional phases of a clinical trial. One other important note is that the FDA really is a friend in this process, and they have provided this timeline of interactions that can and probably should occur with the FDA. And I've heard of some instances of um, attempts to skip the pre-IND meeting for faster translation to the clinic, and then the FDA rejecting an IND submission. And really, in the end, that ends up costing more time and resources to have to repeat those often expensive proof of concept and safety and toxicity studies. So, really interacting with the FDA and getting their feedback as often as possible is really valuable. The FDA also has opportunities to interact directly with foundations through patient listening sessions and also through patient-focused drug development meetings. And so the FDA really is attempting to incorporate the patient voice more in medical product development and regulatory decision making. So these are, again, great ways to interact with the FDA, whether it's through official meetings or through patient-directed meetings. So what we've kind of thought of as the traditional path of moving through this drug development is really a foundation would partner with an academic institution. So they would find a researcher such as myself that works in an academic medical center with a laboratory to perform a lot of this necessary understanding of the disease mechanism and then the preclinical testing and possibly even the toxicity studies. At some point along the pathway, historically, industry has gotten involved to really support the really expensive part of translating a therapy into the clinic with the hope of ending up with an FDA-approved product. There are dozens of companies that are involved with FDA-approved cell and gene therapy products, and there are even more on the landscape than are depicted here in this diagram. However, as was published just a couple of years ago, there's really been unintended challenges in bringing some of these cutting-edge therapies from bench to market, specifically for rare and ultra-rare diseases. And this is a table, again, it's over a year old now, but what they're showing is the number of gene therapy programs that have been licensed to an industry partner and then dropped, meaning that the industry partner for some reason or another, usually business, decided that this one-time treatment for a very small patient population was not commercially viable. When an industry partner drops a program, that really results in lost time and lost resources to get the program to that point of development. So there was an exciting first FDA-approved gene therapy from a nonprofit organization occurred in December of 2025. So this was the Telethon Foundation, where they were able to achieve FDA approval for Wiskott-Aldrich syndrome. And the FDA really commented that they exercised regulatory flexibility to address the unmet need for this rare disease. And we hope that we continue to see some of that regulatory flexibility for rare and ultra rare diseases. So today you're going to hear very shortly from some different foundations and their experiences. So there are some foundations, you're going to hear from Kim Nye about the Test Research Foundation that followed maybe that more traditional path where they partnered with an academic institution. In this case, it was UT Southwestern, and then licensed their gene therapy to a company. In this case, it was Taysha, and they've been navigating this traditional pathway. And so you'll hear a little bit about some of the successes and challenges about this pathway. And then there's other programs. I'm sure many of you know Terry. His son had SPG 50. And while they initially partnered with an academic institution, instead of licensing their program to an industry, Terry actually developed his own not-for-profit called Elpida Therapeutics to help really continue to advance this therapy into additional kids. So this is just showing one way that foundations have really continued to change this traditional path of drug development. And there are other foundations that have attempted to change the path even earlier. And in this case, this organization supported generation of a bioscience incubator. So even starting at the laboratory stage to really help with the development of a gene therapy in the laboratory to help expedite this pathway. Another thing I wanted to just bring to attention that's kind of growing in use is the use of contract research organizations or nonprofit biomedical research institutes. And these sort of institutes can really help with some of the heavy lift from the academic institutions, is the academic institutions often are, limited in some resources and funding, and rely a lot on grants, primarily from the National Institutes of Health. And if we're limited on resources, we can put some of the onus of this research on contract research organizations. However, these CROs or CDMOs or nonprofit research institutes, they still need partners to drive the research. And generally, these companies aren't coming up with the ideas and understanding the mechanism of disease and coming up with a therapeutic strategy. But they can really help with the model generation and or the efficacy testing or the safety testing. Historically, it's been academic partners that to help to drive this research, but increasingly foundations are working directly with CROs. And for example, Fox G1 Research Foundation has partnered with Charles River as a CDMO. And I'm not advertising for any specific organizations. I'm just providing some examples of places where foundations have used this partnership to advance their therapy. Another example is the Jackson Laboratory now has a rare disease translational center, and there are many foundations that have used this resource. Different organizations might specialize in different parts of that pathway. So, for example, some of them might be better at model generation and characterization, where others might be better at the actual manufacturing of your drug product or your safety and tolerability testing. So, depending on what resource is needed at what stage in development, these different research organizations might be of use to different foundations in that pathway of drug development. I did just want to kind of end this part recognizing these barriers for gene therapy development for rare and ultra rare diseases, and that a lot of people are thinking hard about this and trying to find ways to overcome these barriers for these commercially pre-viable gene and cell therapies. So, for example, ASGCT held a workshop in 2025 with getting a lot of people in the room to think through this challenge, and then they even published this review in molecular therapy last year. And ASGCT has now even partnered with an accelerator partner to try to advance, commercialize promising rare disease treatments. So not only has there been a lot of thought about this, but actionable ways to attempt to approve some of the access of these gene therapies for rare diseases. Okay, I'm going to shift gears a little bit and give two real-world examples from programs that I've personally been involved in. One was kind of that more traditional pathway, and one in which worked more with a foundation to veer off the traditional pathway. So the first little story that I want to give is about the development of a gene therapy for a disease called Krabbe disease. And Krabbe disease was really poised for success of a gene therapy because it was identified over 100 years ago. So there's been 100 years of work to understand the disease. The disease mechanism is well understood. We know the enzyme GAL-C that causes Krabbe and the cytotoxic substrate psychosine that builds up in the cells and causes the pathology. Further, we're fortunate to have multiple animal models, including large animal models, as well as naturally occurring mouse models. And there's a standard of care in which patients have been treated with hematopoietic stem cell or bone marrow transplant since the late 90s. also, since the early 2000s, gene therapy has been evaluated in this disease. So when I came on the scene to attempt to develop and translate a therapy for Krabbe disease, it really was poised for success due to this rich history and understanding, and also that it's a single gene defect and the gene fits well inside of an adeno-associated virus vector. Just very briefly, as I mentioned, that the standard of care for these patients was already hematopoietic stem cell transplant. Decades of research has shown that when implemented early, this can really help improve the survival and help the cognitive neurodevelopment of patients. However, it was also known that transplant alone did not fix the gross and fine motor, and it's because the transplant did not target the peripheral nervous system. So our initial therapeutic approach was the addition of an adeno-associated virus gene therapy to the standard of care, the hematopoietic stem cell transplant. So in the laboratory, we used an adeno-associated virus. We knew the disease-causing gene, GAL-C, so we put that inside of the virus and tried it with different promoters that would express the GAL-C in the cells that we needed. Because we really needed to target the peripheral nervous system for our first trial, the delivery route was an intravenous delivery, because we also know that certain serotypes of AAV can get out of the blood, cross the blood-brain barrier, and enter the cells that we need in the blood. With this gene addition therapy, the virus itself is rapidly degraded, but it deposits the gene of interest, which then starts to produce the therapeutic protein. So in this case, GAL-C. We had added advantage with Krabbe disease that it's a lysosomal storage disorder, and this enzyme could even get out of the cells we targeted and cross-correct and treat other cells. So we had our therapeutic strategy, and we had a mouse model that had been around for decades and well characterized. Initial preclinical studies were performed in this mouse model where we were able to show a clear dose response and really improve the survival and outcome in this preclinical model. I was then fortunate, at this point I was at University of Pennsylvania, to have a large animal canine model of Krabbe disease. While I know this is, difficult for many of us who love animals, these animal models are also especially valuable because they mimic many of the signs that are shown in human patients, such as ataxia and tremors. These puppies also have pelvic limb paralysis, and they lose hearing and vision. And these larger animals, we can also mimic many of the biodistribution challenges with gene therapy. And we had, because the disease was progressive, we could tell in a relatively short time of about 4 months if we were able to treat this disease or not. I'm not going to go into any of the scientific details because we published this in Molecular Therapy in 2024. But in short, we repeated what they did in the mouse study, and we also were able to show that we improved survival and that there was a dose response. And in this very valuable large animal model, out until 2 years, we were able to show remarkable improvement of disease in animals that usually don't live beyond 4 months. So this was really powerful data in 2 model systems, including a mouse and a large animal model, in which we were able to establish clear efficacy and a clear dose response. So at this point, we had sufficient data to translate to the clinic, and we're really excited about the first ever gene therapy for Krabbe disease, which is a really devastating disease. So this gene therapy was licensed to a company called Forge. We had the added advantage that Krabbe was then added to newborn screening, and we were able to detect these babies really young and initiate treatment in a really timely fashion. However, last year, Forge decided, after strategic review, to make the difficult decision to discontinue the development of this gene therapy program. And they recognized that this was devastating for the Krabbe community. So it really was difficult for those of us that put years and years into the development of this therapy, and even more so for the Krabbe community, to have a therapy that could be life-altering, then discontinued due to business decisions. So again, it was a disease that was really poised for success. We understood the disease mechanism. We had the animal models. There was a clear therapeutic strategy. We were able to demonstrate efficacy and safety, move this to the clinic, and even throughout all of that success, we still were not able to succeed in getting this all the way to market approval. We are continuing to work on this as an academic endeavor with the second generation gene therapy. So we do have hope for Krabbe for the future. But I want to move to a more recent story, which was a little less traditional, in which many of you know Amber Freed, who started a foundation called SLC6A1 Connect. So myself and Catherine Meyer and a number of other motivated investigators and contributors at Nationwide Children's Hospital partnered with SLC6A1 Connect. And as an academic and foundation partnership, we were able to move through the model development and characterization, therapy development, efficacy, dosing, and safety studies. And we're able to successfully move this to the clinic in 2025. And I hope to be able to share more details of the story in meetings to come. So what I hope today that I've been able to demonstrate is a nice balance of hope and reality in the development of genetic therapies, and how different foundations are coming in at different starting points, and really there's a different pathway for everybody based on where they're starting and what their therapeutic modality is. And hopefully you are aware that there's a lot of free and reliable patient-friendly resources on cell and gene therapy at the ASGCT website. So please explore more of our patient education. We also have an annual meeting, which will be in Boston this May. And if you haven't already, please subscribe to the Patient Press, because there's a lot of great information in this newsletter for patient advocates. And this is my contact information, and I'm happy to take any questions.
All right. So to kick off Dr. Bradbury's Q&A, we want to reveal the responses to our poll to the audience, where our question was, where is your organization currently in its cell or gene therapy development journey? So 9% of our audience are in the learning exploring phase, while the majority, 36%, are in the preclinical research phase. 9% are in the trial planning or safety studies phase, and then 19% are in the active clinical trials phase. So, taking that in mind, Allison, what would be your biggest takeaway or piece of advice for patient groups who are in the preclinical stage in the gene therapy development journey?
Hi, Rachel. Thanks for having me today. It's really great to hear that there's so many engaged people watching this Empowering Patients Summit, and that we really are spanning the very early discovery all the way to the clinical trial stage. I personally live in the preclinical translational step of this development pathway. And so I would say for those foundations, as I mentioned in my presentation, it's really critical to understand the mechanism of your disease so that you can make that safe and targeted gene therapy approach. And really, at that point, you need to make sure you have the correct model systems, which we have some specific questions in the chat that we'll get to, whether those are in vitro and or in vivo model systems, and that you're really trying to develop the most safe and effective gene therapy approach for your specific disease indication. And then you really just have to go through those rigorous and robust preclinical studies, which can be long. So I know that time that those preclinical studies take can be pretty painful for some of these organizations, because we know with a lot of these diseases, time is really critical, and there is an urgency that we need to meet. And then after the really proof of concept, the preclinical studies, then the next really critical step is that safety and tolerability studies to make sure that your therapy is indeed safe before going to first in human.
Fantastic. You mentioned models. Let's discuss those a bit. So one of our questions from the audience is, do you absolutely have to have animal models? Can you skip them completely and use organoids or other methods?
That's a great question. And it really depends on what your treatment modality is. So, for example, if you're going to do an antisense oligonucleotide, you don't necessarily need a disease animal model for each of those, because the reasoning is a lot of the mutations that you're trying to target with an antisense oligonucleotide actually aren't conserved in a mouse between a human. So you don't have to make that animal model because it really wouldn't recapitulate the human disease. Therefore, for some treatment modalities like ASOs, you can actually test them in patient-derived cell culture models. However, once you have your readout and your cell culture models, you do need to do safety testing in animal models still. And the FDA has clear guidance on antisense oligonucleotides to do tolerability studies in rats. If you're looking at that in vivo gene editing, where we, of course, are all familiar with that success story with baby KJ, it was a first in human in vivo gene editing with a lipid nanoparticle. And they did that proof of concept work was in cell culture and then in a mouse model. And then their safety and tolerability studies was done in a non-human primate, so a large animal model. So particularly for first in human, there might need to be really all three types of model systems used. When it comes to AAV gene replacement, if it's a capsid that's already been FDA approved, such as AAV9, then the FDA might permit rodent safety tolerability studies, such as mouse or rat. However, if it's a capsid that's never been in humans before, the FDA will likely require non-human primates or other large animal models. So it really is dependent on the disease and the therapy modality. And the FDA really does help provide guidance on what they expect to see. So those pre-IND meetings can be really informative.
Yeah. And would you say it's also an ever-changing landscape, so that although there may not be historical prior examples for being able to move an AAV vector straight from cells to human, could that be something that maybe in the future the FDA would consider? And
Absolutely. This is definitely a shifting landscape as we talk, and the FDA has recently put out guidance that they'd like to move to more in vitro models and kind of away from requiring more animal models. I'm not aware of anyone making the jump yet from in vitro to in human with a genetic medicine, but that's not to say that won't happen in the future.
yeah, absolutely. So, just to stick a bit on the models, if gene mutation is well conserved in animals such as mouse, do you still need a humanized animal model?
No, a humanized animal model usually isn't needed unless your therapy really needs to target something that's not going to be conserved between the mouse and the human. But if you're trying to just like knock in a patient specific, for example, missense mutation, and then try a gene replacement therapy, we typically don't need to humanize the mouse in that instance.
Okay. And then to pivot a bit, let's talk about different types of disease. So, would you say that gene therapy may only work for certain neurodevelopmental disorders, as, there may be time limitations for when a therapy is provided as to when you would expect benefit? It appears that potentially some of our audience has heard that it only works for neurodegenerative diseases, and that there's doubt that the brain could be improved after it's been developed. If you could comment.
Yeah, this is always, a conundrum with both the neurodegenerative and the neurodevelopmental diseases. And really it's that, like, what is your window of intervention possibility? And they really have kind of unique challenges, and that neurodegenerative diseases, if the disease has progressed so far that really cells are really damaged or have been lost, then you actually might not have enough of those cellular targets for the gene therapy to get into, and for those cells to become the factory and to start producing the protein that you need. So for diseases where it's neurodegenerative, if you intervene too late, there really may not be sufficient benefit. And the contrast with neurodevelopmental disorders, if the disorder really happens very early in the developmental process of the fetus, then the question is, if you intervene once the child is born, is there going to be benefit? And I think it really depends on what the key characteristics and phenotype of the disease are and what is clinically meaningful for that family or that disease group. For example, a lot of neurodevelopmental disorders have a primary seizure phenotype. So if you were able to attenuate that seizure phenotype, would that be clinically meaningful? And I think gene therapy has been able to show some promise. Example diseases like Rett syndrome and Dravet syndrome, where they have a neurodevelopmental phenotype, and being able to really improve some features of the disease, such as seizures, is clinically meaningful. However, the ability of those genetic therapies to be able to improve things such as intellectual disability really might not be there, particularly if the cells didn't develop in the first place. A gene therapy is not going to be able to make cells if they weren't there in a neurodevelopmental case, and it's not going to be able to revive cells in the neurodegenerative case. So it's really having those real expectations of what the genetic medicine is going to do, and if that's going to be clinically meaningful for the disease.
Absolutely. I couldn't say it better myself. So, thank you so much, Allison, for giving us your insight into how researchers pursue certain conditions and treatments and the role patient groups play.